Greetings. We are from G36, and based on our case study, we are tasked to identify sustainable source of protein and processing methods based on separation or reaction engineering knowledge. Hence, we have chosen microalgae as a sustainable source of protein. Protein is a major and versatile constituent of food products. Apart from the nutritive value, the physical, chemical, and the behavioral properties of proteins during processing play a significant role in determining the end quality of food. Due to the structural versatility and amphiphilic nature of proteins, they can interact with other food constituents such as carbohydrates, fats, water, vitamins, minerals, and other proteins through a range of interaction and bonds. The demand for protein ingredients has surged over the last few years. The global protein ingredient market was valued at $38 billion in 2019 and is expected to grow at a rate of 9.1% from 2020 to 2027. Consumption of animal proteins have considerably increased in the recent past, as well as with a growing interest in overall protein, the market for plant protein ingredients is expected to grow significantly.
Factors of driving demand for protein is that it plays a major role in demand through diet, supplement, and food usage. It is also because of the associated health benefit of consuming meat, where increased awareness of the impact of food production with consumption of environmental and health among society. In the context of protein, the negative impact is mainly associated with animal-derived protein, with reports that 12% of GHG emission derived from the livestock production and 30% of human-induced terrestrial biodiversity loss can be attributed to animal production. Furthermore, the driving factor of global demand also include population growth. In addition, socio-economic changes such as rising incomes, increased urbanization, and aging population, whereby the contribution of protein to healthy aging is increasingly recognized.
Microalgae is well known as a viable source of biological components such as carbohydrate, lipid, pigment, vitamin, and polyphenol, especially proteins. The protein quality in microalgae is known to be similar with some of the traditional protein sources like milk, meat, and egg. In general, the protein content of microalgae constitutes a major portion compared to lipid and carbohydrate. The total protein content from microalgae, especially Chlorella species, is about 51 to 58%. There are several advantages of using microalgae as main source of protein. For example, rapid growth rate, high productivity, can withstand harsh conditions, and ability to be cultivated using exhaust industrial gases. With that, it can indirectly reducing greenhouse gas emissions. Three-phase partitioning TPP was introduced as an efficient approach in extracting and purifying enzymes and biomolecules. TPP is easy to operate, efficient, and scalable, where the salt is added to the aqueous solution containing the target product, followed by the addition of T-butanol to form three phases. The top phase of TPP is the T-butanol layer, middle phase is the protein precipitate layer, and bottom phase is the aqueous layer, where the solubility of T-butanol with water will change with the addition of salt.
Now we will proceed to the next part, which is methodology. The chemical reagents used in this proposed method are T-butanol, ammonium sulfate, sodium sulfate, magnesium sulfate, magnesium acetate, dipotassium hydrogen phosphate, Bradford reagent, and tri-HCl buffer. The microalgae selected is the green microalgae, C. vulgaris, strain FSPE. BG11 medium was used for pre-culturing the selected microalgae for around 7 days, and then the microalgae was cultivated in batch mode for 10 to 14 days by supplying 5% of carbon dioxide continuously. The microalgae were cultivated in batch mode using a 1 liter indoor glass vessel photoreactor (PBR) by inoculation from the pre-culture cultivated in a PBR. Light source was provided continuously to illuminate the culture by mounting external LED light on both sides of the PBR. The initial culturing of microalgae and transfer of inoculum from the agar plate to the PBR were performed under a UV laminar flow chamber to prevent biological contamination. The biomass productivity, nitrate content, and pH value of the microalgae culture was monitored during the batch cultivation. The biomass was harvested when the growth of microalgae achieved a stationary phase. The harvested biomass was centrifuged at 6,000 rpm using a centrifuge for 5 minutes to remove liquid content. The remaining biomass paste was frozen at -20 degrees for 24 hours before freeze dryer prior to the extraction experiment.
The study of TPP was conducted with a total working volume of 10 ml using the predetermined initial condition. The system was prepared using 5 ml of 30% saturation ammonium sulfate, 5 ml of pure T-butanol, and 0.5 weightage percentage of free-dried microalgae biomass. Firstly, the microalgae biomass was dissolved in deionized water before mixing with salt solution. 5 ml of T-butanol was then added to a mixture of microalgae biomass and salt solution in the small beaker. The mixture was stirred using the magnetic stirrer at 200 rpm for 1 hour and was allowed to separate for 30 minutes at room temperature. 3 phases were observed and separated carefully by pipetting them out of the beaker. The intermediate protein precipitate was dissolved in appropriate amount of trice hydrochloride buffer and analyzed for protein content. The separation efficiency and yield of protein in all 3 phases were analyzed and compared with the ultrasound-assisted 3-phase partitioning process. All the experiments were repeated 3 times and the mean of the value were reported.
Greetings, I'm Harshni and I would like to present about determining the protein content in microalgae. Separation efficiency of the extracted protein was calculated using equation where VT, VV is the volume of top, middle and bottom phase, whereas CT, CM and CB are concentration of top, middle and bottom phase respectively. The total protein content in microalgae biomass PP was calculated using the equation where NEA is the total nitrogen percentage in microalgae, which is 4.18%, and NTP, which is constant value of nitrogen to protein conversion factor, which is 4.78. Therefore, the final value of PP used in this work is 19.98%. The elemental composition of microalgae is 49.32% of carbon, 8.24% of hydrogen, 4.18% of nitrogen, and 0.15% of sulfur. Yield of proteins from microalgae biomass is calculated using this equation, where Wi is the initial weight of biomass microalgae used in the study. Based on an article, the data of TPP method obtained shows that the yield is 25.15% and separation efficiency of 49.78%. The proteins favorably partition to the intermediate phase and less proteins partition to the top and bottom phase. These precipitate proteins formed in between the top and bottom phases created the 3-phase function. Next, 3-phase partitioning method is compared to conventional separation to prove the efficiency of this sustainable method. TPP has a simple operation with short processing time. It can be performed at room temperature. It also achieves high purity levels. It is non-expensive and feasible for large-scale production. Whereas for conventional separation, it consists of multiple unit operation. It also requires high consumption of toxic organic solvents. It has multiple processing stages contributing to more time consumption and product loss. It has lower concentrations of end products. The conclusion is proteins from C. vulgaris FSPE is successfully extracted using sustainable bioprocessing technique, which is 3-phase partitioning. TPP is identified and discussed as a sustainable method to move towards the realization of producing low-cost nutritional food. These are the references that we used to conduct our case study. That's all from us. Thank you and stay safe.